Welcome to the Wednesday afternoon lecture series, the most important lecture series at the NIH. And welcome to the new year at the NIH. I'm Lou Stout. I'm Chief of the Lymphoid Malignancy Branch and Director of the Center for Cancer Genomics at the National Cancer Institute. And it's really a great pleasure to introduce Dr. Andrea Califano to you. I've known Andrea because of a joint interest in both genomics and early on his interest in B-cell lymphomas, now interested in all cancers. And he's going to be discussing systems biology network-based methods for identifying therapeutic targets. I think his focus is to use these genomic systems biology ways to actually do new clinical trials, which he'll tell us about. Our speaker is extremely well known within the cancer community and more generally in systems biology community. He is the Clyde and Helen Wu Professor of Chemical and Systems Biology, Chair of the Department of Systems Biology and Director of the J.P. Sulzberger Columbia Genome Center at Columbia University Medical Center. And so he brings a lot of interesting background, originally a physicist by training, and he's taken, in my view, quite innovative approaches to identifying molecular factors involved in cancer progression, cancer phenotypes in general, drug response and resistance, and more recently, I think, as he'll tell you about today, using single cell data from cancer patients to predict pathway activation. This involves a lot of math, which Andrea is very good at, and it involves integrating into network models to make a total model of the behavior of at least parts of the cancer cell. He's shown, I think, probably his major contribution, in my view, to cancer biology literature is the idea of the master regulator, which is basically, these are nodes with regulatory nodes within cancer cells that he can identify computationally that integrate a lot of signals and then have output to many downstream targets, and that these are called master regulators, that if you interfere with these, then they have such a broad effect on the cancer cell that typically that induces cell death or a major change in phenotype. And this then leads obviously to a new set of therapeutic targets in cancer that can be addressed by a number of means pharmacologically, and now maybe many of these are transcription factors, as you may hear, and so they may require things like PROTACs to be done to be targeted. So just a little bit of more detail. His undergraduate degree and PhD was in physics at the University of Florence in Italy. His PhD was on the study of chaotic behavior in high dimensional dynamical systems. And having read the book Chaos, I think this does not mean that his dissertation was chaotic, but it is a type of broad analysis of complex behavior of dynamical systems that I think is a good way to think about how biological systems might actually work. He then moved on to postdoctoral training in Florence and then at MIT, and finally spent quite some years at the IBM Watson Research Center in the 1980s, becoming a staff member there, and then moved on to Columbia back into full academia, shall we say, although I think he was doing academics at the Watson Research Center. He moved in 2003 and eventually became professor of biomedical informatics. And now, as I mentioned, director of this Sulzberger Columbia Genome Center. He's a very energetic guy, if you get to know him, and he's founded several companies such as First Genetic Trust and Therasis. He's got many honors, very rightfully, in my opinion, has been awarded the NCI Outstanding Investigator Award. He received the Ruth Siegel Award in Pancreatic Cancer Research. He's a member of the IBM Academy of Technology and notably of the National Academy of Medicine. The title of Andrea's talk today is Elucidation and Pharmacological Targeting of Non-Oncogene Dependencies at the Single Cell Level. Andrea, it's a pleasure to have you come to the NIH, and I'm always excited to learn from your lectures. So we're really looking forward to your presentation. Thanks a lot.
Thank you, Lou, for the wonderful interaction. I also want to thank you and Michael Gottesman for inviting me to give this lecture. I think the invitation came in March 2020, and then we know what happened. So I'm really glad that we're finally able to do it, even if not in person. The reason I have these logos here is because my entire field would not have developed cancer system biology without these tremendous efforts by the NCI and by the NIH. And I think this is really merits a moment of reflection on how the NCI can really foster the kind of studies that I'll be discussing today. You've seen my my conflict of interest already. This is the latest company that I started in 2016, Darwin Health, and licensed some of the algorithms that I'll be discussing today. So it all started from what we have come to call the cancer paradox. And this, we published it in a perspective in Nature Reviews Cancer. And basically the paradox states something quite obvious that a lot of us have known for ages. But the difference here is that we actually try to find a potential computational solution to address this problem. So the paradox is the fact that if you look at the transcriptional state of cancer cells, tumors break down into subtypes that are so tightly regulated and similar, where the samples are similar to each other, that they're no more different than equivalent number of samples taken from normal tissues of the same number of individuals. And so the reviewers of that perspective didn't believe that statement, so they asked us to generate, and I'm just showing you 4 tumor types. We did it for all tumor types in TCG, of course, and you can see here that blue is tumor, red is normal tissue, and of the matched organ. You can see basically that the distribution looked very similar. And yet, so transcription, these cells are very, very similar. And yet at the genetic level, the mutations are literally all over the place. So that, this is in luminal breast cancer, sorry, in triple negative breast cancer, there's really no 2 patients that have the same set of mutations. And so we argued for a while on how we could potentially find a solution to this problem that would explain why that is. And we went back to Cannon and Waddington in terms of this concept of canonization. Thought that one possible way in which this could be happening is if all the mutations occur upstream of a piece of machinery that in cancer we call the tumor checkpoint, and in non-cancer tissue we call the regulatory checkpoint modules, where a very small number of proteins that, as Lou mentioned, we dubbed master regulators, are actually integrating the aberrant signals that come both from the mutations, but also from both so the distal and tumor microenvironment related signals. And then these proteins essentially are incredibly tightly autoregulated, and therefore their stability is determined by their interactions, and together they regulate the transcriptional state of the cancer cell. And so this paradigm, which are called an oncotecture, basically states that what is really recurrent in cancer is not that much the mutations, but rather, and this is not just in cancer, but also in common disease, but rather the architecture, the signaling and transcriptional architecture that is responsible for their integration and their translation into a transcriptional state of the cell. And so we've published extensively on these points, and basically used algorithms to make prediction, and then going all to the lab and making experimental validation assays that really demonstrated that this paradigm is actually holds together in pretty much every tumor type that we've looked at the molecular level, and sort of mechanistic. And I think I'll try to explain what we call this a kind of a quasi-mechanistic model. But the more important thing is that these studies have now yielded quite a large number of clinical trials and clinical studies with preclinical endpoints that are currently running. Some of them are completed, and I'll show you the results, and others are being planned and will start imminently. They're not presented here, but they'll start in the next few months. So one nice thing about having a model, having this architecture, is that we can now start developing algorithms that are going to first reconstruct this architecture, and we know that it's different in a way that is completely context dependent. So the architecture of a B cell is very different from the architecture of a glioma cells. And so we use this methodology, and all of them have been extensively validated experimentally. All of them have a 70 to 80% validation rate in the lab, in the experimental lab. And the first one is an algorithm that allows you to turn a large number of gene expression profiles into a fairly accurate map, and as a fairly accurate, about 70% validation rate of the underlying transcriptional regulation mechanics. Then this algorithm called VIPRE essentially uses the interactomes that are built by Arachne to then measure quite precisely the transcriptional activity of proteins. Mindy and Digit are algorithms that allow you to go upstream of the master regulator in the upper part of that hourglass model, and to identify quite effectively all the proteins that have a potential modulatory role, where basically their activity or abundance can modulate the ability of one of these master regulators to regulate its targets. And then finally, PrEP, it was the first algorithm developing in collaboration with Barry Honig to actually predict protein-protein interaction using structural biology data. So let me spend a couple of minutes on VIPRE, because it's the workhorse of the lab, and a lot of the things that I'll show you are based on it. So if I don't explain this in a way that is understood, then you may miss the rest of the talk. So the way VIPRE works is akin to a highly multiplex gene reporter assay. So what we basically do, if you want to measure the activity of, say, MYC in a B cell, what you can do is you fuse a TERT promoter with a luciferase, and then you can measure luminescence. And if you have luminescence, since TERT responds to MYC, then you think MYC is activated. If you see no luminescence, MYC is inactivated. That works well if MYC is the only thing that moves in the cell. And obviously, because the TERT promoter is highly pleiotropic and responds to other transcriptional regulators, it will not work well if everything moves. And so what we tried to do is to generalize this concept of a gene reporter assay, so that we now can use about 100 targets of every regulatory proteins, both positively regulated and repressed, as the reporter assay. And so from the expression of these targets, actually from the differential expression of these targets in two different states of the cell, we can measure the differential activity of any regulatory proteins in those two contexts. So for instance, you could use two isogenic contexts, one with an oncogene mutation, one with a wild type. You could use two different disease stages or progression stages. You could use cells that are drug sensitive versus drug resistant. And we've done all these studies and published them. And in every single case, we've been able to identify the master regulators and then validate them experimentally. But also, as you'll see, even in very early papers, we showed that we can actually reprogram the state of the cell by targeting the master regulators, because these are essentially the proteins that mechanistically, I should say quasi-mechanistically, because only 70% of their targets are correct, but that mechanistically regulate the state of the cell. So there are three hypotheses, main hypotheses, that stem from this architecture, sort of a major hypothesis that we have postulated. The first one is that master regulators form these tumor checkpoints that in cancer, or we call them regulatory checkpoints when it's not in cancer, and these are responsible for homeostatically maintaining tumor cell state. Homeostatically means that they can essentially adapt to any perturbation and maintain the state largely independent of these perturbations. And these are papers where that has been shown. There's more, but I'm just showing some examples. The second one is that the master regulators in these tumor checkpoint modules canalize the effect of most of the functional mutation event. And again, these are papers. The first one is actually in the LBCL, and so probably brings up memories to Lou. The third one is that master regulators in tumor checkpoint modules represent a novel class of pharmacologically actionable non-oncogene dependency. In fact, these genes are almost never differentially expressed, and the proteins that and almost never harbor mutation that turn them into essentially aberrantly active proteins because of the mutation. So let me show you a case study in pancreatic cancer. This is a collaboration with Dave Tuveson, Ken Olive, Fayyad Nora, and Carlo Maurer, and many others, and has been led by Pasquale Leis and Mikko Turunen in my lab. And see, two of the hypotheses are essentially discussed and validated here. Pancreatic cancer has been challenging to classify and structure because if you look at 3 different studies that were done from gene expression profiles, the cluster that were generated essentially had almost no overlap. In fact, they have no overlap between the 3 of them. So you can see here that according to 4 different studies in tumor and stroma or in the tumor cells, there were 0 overlapping genes that would determine the identity of the subtypes identified by each study. And we thought that maybe this was because, one, there is tremendous stromal infiltration in pancreatic cancer. In fact, sometimes the tumor cells are actually a minority or even a small minority of all the cells in the tumor. And number 2, because within the tumor itself, there may be multiple compartments of cells that have strikingly distinct molecular identities. And so to do this, we resorted to study pancreatic cancer at a single cell level. And I want to show you what the challenge is when you go to single cells. So this is data that was published recently in a Cell paper on kidney clear cell carcinoma, collaboration with Chuck Drake and with a very talented MD PhD students in our labs, Alex Obradovich, where basically we took single cells from these tumors, FACS sorted the CD45 positive compartment, and then perform a analysis using either a CyTech, which uses a panel of 19 myeloid and 19 lymphoid antibodies. I'm only showing the lymphoid here. You can read the paper for the other ones. They basically tell the same story. And those identified 4 populations that you would expect to find in these cells. However, if you take the same cells and now send them to a chromium and you look for the genes that encode for these proteins, you see absolutely nothing. Essentially, there's complete loss of the mRNAs that encode for those proteins, and therefore you cannot cluster do anything. However, when you now run VIPRE on these data, not only you can reconstitute basically everything that was found by antibodies, but in fact you get dramatically increased stability. So your standard deviation versus the mean becomes dramatically better within the subtypes. So let's say CD4 in this case for so FOXP3 for Tregs here, which you can't see at the antibody level, CD14 for monocyte and macrophages, which again, very noisy. You can't see it in the antibody, CD137, which is the IL-7 receptor for the CD4 cells, which is very noisy again. And there are also things like CD19, which we didn't detect well, but this just means that we have to refine our regulons. And in fact, some of the proposal that we have for the next few years address that question experimentally by combining Arachne with single cell silencing data. So basically, we can now use VIPRE to turn single cell data almost like if it were bulk tissue. And so we use data from a prior publication with Dave Tuveson, where we'd actually focus on the fibroblasts to identify these multiple different subtype of fibroblasts that are immunosuppressive. And instead of focusing on the fibroblasts, you focus on the epithelial compartment. You can see how small it is compared to all the cells that we could identify. We validated by inverse CNV that these were actually tumor cells, and then perform our analysis using VIPRE and cluster, but the clustering was done not by gene expression profiling, but rather by protein activity. Why is that important? Because if you did the clustering by gene expression profiles, you would not see these 3 populations. In fact, you would see there's 4 of them. So protein activity generated 4 subtypes of molecularly distinct subpopulation of single cells with a very strong silhouette score that is dramatically improving over a gene expression. And you can see here that we did an additional analysis. So these are the different colors represent the 4 subclusters, but you can see that within this cluster of green cells, there were actually 3 peaks. And so we now do this peak analysis because essentially from the Boltzmann distribution, a peak in the probability density of the cell corresponds to a stable state. And the stable state are very important because the cells that are not under peaks are essentially moving very rapidly from one stable state to another, but they eventually will get there or they have to start from 1. The ones in stable state tend to persist for a long period of time. So those are the cells that you really want to target pharmacologically. And so these are cells that we call them, as you'll see from the next slide, lineage and morphogenic. These are differentiated progenitor state. And this is a very interesting, acinar-like progenitor state. And when you look at the pseudo trajectory, you can see here that essentially you have lineage and APS and MOS converging on this differentiated progenitor state. And this is consistent with the view by Cytotrace and with measure entropy and therefore the stemness of this population, with the APS being the most stem-like, but LIN and MOS also being quite more stem-like than DPS. We then look at the presence of these population within different patients, and you can see here that essentially all the patients presented at least 3 of the 4, and in some cases all 4 of them. The APS are more rare, consistent with the fact that probably a more stem-like population. So in some patients, maybe we haven't seen this simply because we didn't sequence enough cells. But then more importantly, if you look at the biology of these cells, you can see that there are really markers that are highly distinct. These are markers that you can actually use to fax the cells and divide the subpopulation that distinguish these 4 different subpopulations. And just as a summary here, you can see that this population here, a very, very small number of cells, is actually extremely distinct. And these are 3 of these markers are classical markers of ADM, acinar to ductal metaplasia, including SPP1. You can see here represented only in this cell and in these cells. The one cut, which you can see here, is basically activated only in these cells, and the SOX9, which is activated in these cells and these cells. So this analysis really allows you to discover biology that can then be validated that you would have completely missed if you were to use gene expression profile. We then went to other 2 cohorts of single cells, and basically it's the PENG data set and the NOTA data set, and we call it CSY. And you can see that single cells from this data set completely validated the structure in the 4 clusters with the differentiated progenitor cells forming typically the majority of the cells, and then lineage morphogenic and APS cells representing the rest. And this was very helpful because in every single study that we have done, when we do this analysis at the protein level as opposed to gene expression level, we find tremendous consistency in different databases. So these were 35 patients, 21,000 single cells. So then we went to the bulk cohort, and we asked whether we could recapitulate some of these molecular distinct subtypes. And so if you actually just do stratification by bulk profile based protein activity, you see that both ICGC, which is the green, and TCGA stratified according to these proteins here and to these proteins here. And again, these were incredibly enriched in the master regulators of the lineage and morphogenic subtype. However, the APS and DPS subtypes simply would not be identified in any of the data sets. And this is another data set that was generated by us by laser microdissection, collaboration with Ken Olive, of about 200 pancreatic cancer patients. This is the NOTA data set from the bulk. And basically, you can see that the proteins that you find in these data sets as differentiating these 2 subtypes, which are essentially the lineage and morphogenic, are incredibly overlapping in terms of their master regulators. So again, not only on the single cell, but even in bulk profile, now instead of finding gene expression panels that have no overlap, now you find master regulator panels that are virtually identical, highly conserved across these different tumors. And so we went to cell lines because you need cell lines to do some of the experiments. And so we identified that in a collection of 8 different cell lines, we could find you can find single cell representative lineage, DPS, APS, and morphogenic subtypes. And this gives you the identity of the line. So for instance, KPON1 was predominantly lineage and DPS. You can see it essentially along this axis, while for instance, KP4, very aggressive cell line, was mostly morphogenic. This lineage and DPS, this is morphogenic and DPS. And then we did lineage, barcode-based lineage tracing assays because we wanted to know whether the plot that I showed you before based on entropy was actually really holding, or whether there was also de-differentiation. So whether the plasticity was going 2 ways or only 1 way. So this is in KP4 cell. We now done it also for PA2 cells that are the lineage-based. These are the morphogenic ones. You can see that these are barcodes that you see at time point 1, which is about 48 hours after the start. and only in this population. And now you can see that at time point 2, which is about 25 days, you see that these barcodes are reprogrammed to the DPS cells. And so obviously we can see there is tremendous plasticity this way. But when you actually look at the opposite, these barcodes that were in the DPS cells and then are found in the morphogenic cells, you also see that. And so this shows that these cells are really spontaneously reprogramming between these two states, albeit with potentially slightly different kinetics. And so we then move to selecting the 3 most morphogenic and the 3 most lineage cells from a large collection of pancreatic cancer cells. You can see here the 3 that are morphogenic and the 3 that are more lineage-like. And then we perform CRISPR-I-based silencing with the library of all the transcriptional regulators and co-transcription factors and negative control, of course. And then we use this library to look at 2 things. The first one, we asked, are there master regulators that have that elicit essentiality that there is actually subtype specific? And so we generated a signature to compare the lineage versus morphogenic cells, and we asked whether they were enriched. We did both by knockout and by CRISPR-I, and you can see really striking enrichment, 10 to the minus 6 in terms of the p-value of the enrichment of the predicted master regulators in one lineage versus the other. And then we actually ask, are there some common dependencies that can be found by, for instance, comparing both lineage and morphogenic cells to their normal pancreatic tissue counterpart in GTEC. And so this is a signature or essentially of cancer progression. And again, there was really a striking enrichment of the top master regulators in genes that are essential in all the 6 cell lines, as opposed to just one subtype versus the other. So this basically, I think, supports the hypothesis that master regulators are a new class of non-oncogene dependencies. They're very enriched. I'll give you more data on that. But we also published a lot of other papers where this has been shown. The more interesting things is to actually show that these genes, these proteins mechanistically control the state of the cell. To do that, we ectopically express the master regulators of the lineage subtype in KP4 cells, which are strongly morphogenic. So this is the signature these are the activity of the proteins in the KP4 cells on the signature that distinguishes morphogenic versus lineage. And this is the signature of the lineage subtypes. And so you can see here that almost every ectopic expression of virtually every master regulators induce significant activation of the proteins that were inhibited down activated in the morphogenic subtypes, but they had a mixed result on the proteins that were activated. But one of them, which is OL2, actually induced almost complete reprogramming. So these cells after induction look exactly like a lineage cell, no longer as morphogenic cell. And then using this data, because we actually had the RNA-seq after each one of these ectopic expressions, you can actually reconstruct the full architecture of the master regulator modules, and you now understand mechanistically why OVL2 is responsible for driving that transition, because OVL2 actually represses all the master regulators, or a large number of the master regulators of the morphogenic lineage, and activate a large number of master regulators of the lineage. And the ones that it also regulates indirectly regulates the ones that are missing. So taken together, this induces really dramatic reprogramming. We then ask, is this actually due to we didn't see the reprogramming because the effect occur only in a subset of the cells. When you reprogram, sometimes you have low efficiency. And so we repeated these assays at the single cell level, and we overexpressed genes with an MOI of 1, so that some cell received more than one cDNA. And so you can see here that essentially a lot of different combination and single genes actually statistically significantly reprogram the cell, but with different efficacy and efficiency. So OVL2 is here, and first when it's combined with HNF1A, it dramatically increases the efficacy of the reprogramming. And then basically anything that you see with stars reprograms. And this is just a subset of the cells that we confidently call reprogrammed to the lineage subtype. So this, I hope, demonstrates that these proteins are responsible really for homeostatically maintaining the state of these cells, and by genetically interfering with them, you can reprogram cell as you want. So we then extended, this is a large study that was published in Cell about eight months ago, where we essentially asked, and we generalized these to all tumors. Can we use the oncotexture hypothesis to really look at tumors one sample at a time, ignoring anything we know about subtypes, anything we know about tumor type, et cetera, other than in using, obviously, the right regulatory networks from a tissue that is matched. And so this is in collaboration with Corey Barishen and Alvaro Aides, which was now an independent investigator at Barcelona, and Evan, Paul, and Sunny from my lab, Mariana, who's the chief scientific officer at Darwin, who started this project when he was still in the lab. And so what you see here is that instead of doing what we did in a previous Cell paper to find the mutation upstream of the master regulator, which was going from the gene expression to the master regulators using VIPRE, and then going upstream of the master regulators using an algorithm called DIGIT to basically identify all the possible mutation that can affect the activity of the master regulators. What we did here, we put the master regulators in the crosshatch of both the upstream genetics and the downstream transcriptional regulation. And this allowed us to identify, to stratify the entirety of TCGA, one sample at a time, in 112 tumor subtypes. And the first striking things was that when you look at each one of the tumor type, all of them except CoAD, which was just borderline not significant, identified subtypes that had statistically significant differential outcome. And this has been very challenging to do by gene expression. For instance, in prostate cancer, I would challenge you to find any expression-based clustering algorithm that identifies in TCGA subtypes that have different outcome. This is an example for kidney cancer where we found 5 subtypes, and you look at S3 versus 5, really striking differential outcome. The other point is that when you compare the stratification you can do by protein activity, these are the blue clusters, the blue Violin plots. This is the silhouette score that tells you how tight these clusters are. The red line, the dotted line, is basically the line for statistical significance. So anything above is significant, anything below is doubtful at best. And the red Violin plot are what you get from unsupervised gene expression analysis. So the clustering of TCGA by gene expression analysis really does not produce clusters that are very tight. So this is kind of the money slides in terms of showing that the genetic is upstream of the master regulators, because basically we asked a question, and we asked this question one sample at a time. How many of the genetic alteration that we can identify in that sample by GSTIC2 and CASM can actually be placed upstream of the master regulators of that sample? And what you're seeing here, each different color line is one of the 112 subtypes. And of course, in some tumors like ovarian cancer, where there's an enormous number of passenger mutation and structural events, the saturation was not very fast. But in AML, for instance, you get to explain about 100% of the mutation with somewhere around 15 to 25 master regulators. And so this basically is a saturation curve, tells you how much of the genetics is upstream of how many of the master regulators. This slide is very important because it tells us that when we want to really kill a tumor, and we want to do this by targeting the master regulators that are aberrantly activated as a result of the mutations in that samples, we really have to focus on average of the top 25 most activated and the 25 most inactivated. Importantly, these analyses identify only 407 master regulators, and we validated that 141 of them in the Achilles project are actually subtype essential. That's about 35%, which is a pretty striking enrichment. So now I want to tell you how we target these things in the clinic before showing you how we do it at a single cell level. And so, this is a large collaboration that's been going on for 7 years with Andrew Kuhn's lab. Also, Mariano Alvarez started this in the lab, and now Prab Mundi in my lab and Phil De La Cruz in Andrew's lab are pursuing it. So this is based on 2 algorithms. The first one, both of them are actually New York and California Department of Health approved and CLIA compliant. The first one is a really very simple-minded, adaptation and extension of the oncogene addiction postulate that is actually tumor mutation agnostic. How's it agnostic? Because all we do is ranking all the proteins in a tumor from the most inactivated to the most aberrantly activated, and then we walk down this list until we hit one for which a high affinity pharmacological inhibitor already exists. And so, for instance, for inflammatory breast cancer, about 20% of metastatic breast cancer, we identify HDAC6 as one such dependency. Recolinostat, at the time the study was initiated, was a very high affinity inhibitor just specifically for HDAC6. And so we started a clinical trial in collaboration with Jose Silva and Jiang Yu, who is now at St. Jude and really came from my lab in breast cancer. And what you're seeing here was really quite striking. Basically, every single patient that responded actually had higher aberrant activity of HDAC6 computed by VIPRE analysis compared to the patient that did not respond. So you can see there's quite a significant response in the responder versus non-responders, and the area under the curve was essentially 1. So this tells you that, you know, this is not a study that's going to save people's life necessarily, because obviously in the end you'll see recurrence because you're targeting only one protein. But at least it tells you that we can predict with very high accuracy who will respond clinically, not just in a PDX model or in a cell line. So this is on medRxiv, and it's in review. So then we extended this concept in collaboration with Andrew Kang, and basically now we've done this for every cohort, not only in TCGA, but in Target, in ICGC, in every single cohort we can think of. Essentially, we rank every protein that is druggable in terms of what is the cohort in which it is the most aberrantly activated one. And so, for instance, we found that exportin-1, which is a target of selinexor and eltanexor, is actually really aberrantly activated in Wilm tumors and malignant rhabdoid tumors and germ cell tumors. In fact, more so than in any other tumor cohort. And so you can see that the sensitivity, the IC50, is in the 10 to the minus 8 micromolar range. So we're talking picomolar range. These tumors, when transplanted, responded strikingly well with essentially complete response to the drug, and then showing relapse when you stopped administering the drug. And so, you know, these are patients that have been treated based on these findings. This is a 70-year-old patient that had a recurrent Wilm tumors that essentially failed to respond to pretty much any therapy they'd thrown at it, had numerous metastatic deposits in the peritoneal cavity, had refused to get additional chemotoxic therapy because of side effects. He was put on selinexor and essentially has been doing spectacularly well. There's been no detectable lesion after his last surgery, and is back in school and playing soccer. This is an ultra-rare tumor called calcified nested stromal epithelial tumor. There's only 40 reported cases of this tumor, not per year in total. A patient, a 14-year-old patient, presented with this tumor, failed to respond to every possible therapy, including a therapy meant for Wilm's tumors that did absolutely nothing for him. And OncoTarget found that PDGFR beta was the most aberrantly activated protein, and sunitinib, which is actually well tolerated in patients, was then selected by the oncologist to treat the patient on memorial. And you can see really the striking response at the level of the imaging. So now let me talk a little bit about the more sophisticated approach. So I told you there's 25 master regulators aberrantly activated, 25 master regulators that are aberrantly inactivated that explain the genetics of a sample. Wouldn't it be wonderful if we could find a drug that actually invert their activity? And so basically, can we do drug perturbation in cells that recapitulate the aberrant activity of these master regulators with a large range of drugs, and then find the drugs that is able to completely switch the activity? Now, this sounds insane because it's hard enough to find a drug that targets one protein, let alone 50. But the reason this actually works, and it works quite well, this is a Nature Genetics paper in which we showed the inversion of master regulator activity in neuroendocrine tumors of the gut by a drug called entinostat, is that these, as I showed you for pancreatic cancer, these master regulators really work as a module, and therefore they all regulate each other. So when you hit the right entry point, even in a way that you don't necessarily understand, the entire module literally switches off. And that module is what controls the state of the cell, and so you expect the tumor either to reprogram or to die. And it's very important to select models that are actually matched to the master regulators, otherwise you get complete junk, but we've shown this in several papers that we've published. So we've now generated this really remarkable repository of drug perturbation profiles. These are genome-wide, so in comparison to CMAP, they're not using 1,000 landmark gene, but they're doing genome-wide. They're done in cell lines that have been matched to the largest number of patients in TCGA based on conservation of master regulators. And the perturbation was done at the concentration, at the 48-hour IC20 concentrations, basically the highest sublethal concentration of the drug you could achieve, so that you don't have confounding factor or confounding event from cell stress sigma pathways or cell death pathways. And so this repertoire can be now used to make oncotreat predictions in virtually any patient. So this is the N-of-1 study, again, 7 years in the works, where we've enrolled about 130 patients across 18 different malignancy, transplanted them in PDX models, generated 45 PDX models, and we have tested in independent drug arms in low passage PDXs, 34 different drugs predicted by either oncotreat or oncotarget. And so these drugs were predicted either because they were targeting a single master regulator or because they were targeting and inducing inversion of the entire master regulator signature. So I think these are pretty striking results. You're seeing that these tumors were selected specifically in the enrollment criteria to have failed 3 to 7 lines of therapy, essentially considered to be untreatable. You can see on DMSO, they double in volume, all of them double in volume size in less than 30 days. However, in 16 oncotarget drug arms and 18 oncotreat drug arms, 8 of which were predicted by both methodologies, you can see that oncotarget induced a response in about 60% or more of these PDX models, and oncotreat in more than 90%. So this was very encouraging, but we also want to know, well, what about using conventional therapy as opposed to vehicle control? And so we did a test where we tested randomly selected tyrosine kinase inhibitors and compared into DMSO, and you can see here there is absolutely no response. And this was done in 13 negative control arms. So this, in our opinion, is the first study where the results of a study that predicts activity and sensitivity of a drug in vivo de novo is actually validated in a way that is statistically significant. And you can see here pancreatic cancer, these are the 5 drugs that we predicted, and you can see that 3 of them actually had pretty significant effect, partial response or stable disease. One of them was borderline, only one failed to respond in the mouse. The more interesting thing is that by taking samples at 3 hours after the third dose of the drugs, which are basically pharmacodynamic samples, we could validate that in every single case out of the 18 drugs that we tested with oncotreat, so 15 out of 18 of the treatment induced exactly the reversal of master regulator activity in vivo that we had predicted from cell lines. And so this is very important because it validates the fact that when we match these cell lines with the algorithm called OncoMatch, we actually find models that are relevant to the in vivo study. And we don't care at all whether the drugs will kill the cell lines, because that's not happening. But we care about understanding the drug mechanism of action, because then we can match it to the mechanism of action of the tumor in the patient. So let me show you how this can be moved to single cells. In single cells, we studied this in collaboration with Jeremy Worley, who's now BMS, and several other investigators, by focusing on a population of tumor-initiating cells that can be enriched for by sorting using CD49F and EpCAM. And this population, some people call it enriched for cancer stem cells. We don't like the terminology. We call them stem-like progenitor cells. And so we sorted about 75% of the cells isolated from 7 breast cancer metastatic patients, including 5 hormone positive and 2 triple negative. And then 25% we just broadly gated on EpCAM so that we get also the differentiated cells. We did a VIPRE analysis. We predicted the drugs for the different subtypes that we identified, treated in vivo, and then assessed whether the drug did what they were supposed to do. That is, deplete one population but not the other. And so what you see here is really striking because, first of all, if you do gene expression-based clustering, you see that every patient forms a different clusters. And so people call this a batch effect. This is not batch effect. This is biology. And the biology comes from the large copy number alteration that are present in individual patient, which contribute some difference to the gene expression signature. But at the level of the protein activity, the cell buffers those differences. And so only the differences that are important in establishing the state of the cell as a transformed cell are retained. And so what happens is that when you go from gene expression to VIPRE activity, now all the patient overlap. And that requires no normalization, no batch effect correction. And what you're seeing now is the difference between the top and the bottom is the hormone positive versus the triple negative. And so basically you're retaining the important biology and you're missing out the things that are just alteration that are not functionally relevant. And also you see that when you go left to right, you see striking enrichment in markers that have been published and reported as established markers of stemness in tumor initiation like BMI1, Notch1, KLF4, SAL4, et cetera. And in fact, by cytotrace analysis, which looks at the entropy, you can see really strikingly high entropy at this end and really differentiated cells with very low entropy at this end. So what we did, we took the 10 most stem-like cells from each patient and compared them to the 10 most differentiated cells from the same patient. And we did this independently across each patient. And then we integrated the results. And lo and behold, the master regulators were exactly the same across the patient. So the integration was very, very successful. And then we use OncoTreat to predict the drugs using perturbation in BT20 cell lines, which actually really had a strong conservation for the activity of these master regulators. And what was surprising is that two drugs emerged, ivermectin and albendazole. These are anti-helminthic drugs that are typically never used in cancer, but came up really, albendazole is the number one most statistically significant. And so we treated in vivo these mice that were transplanted with the tumor biopsy for a triple negative, very aggressive patient with ivermectin, albendazole, paclitaxel, and vehicle control, dissociated the cell, profiled them, and then sequenced them. And what you're seeing here is that we measure the enrichment, and so increase in density versus decrease in density by comparing each treatment to the vehicle control. So as you expect, when you treat with paclitaxel, which is known to target differentiated proliferative cells, you see striking depletion of the proliferative cells, which are in blue, sort of the cooler tones of the color spectrum, and dramatic replenishment of the stem-like compartment. So these tumors stop growing because you're killing a large amount of the cells, but they accumulate a lot of cells that can restart and reboot the tumor. However, with ivermectin, and even better, dramatically better with albendazole, you see that there was really striking depletion of the cells that are more stem-like, and in fact, increase in the cells that are in the population of differentiated and proliferative cells. And so we thought maybe what we should do is use these two drugs in combination because they're targeting two different populations, maybe they synergize. And indeed, we did this preclinical study with Andrew Kang, and you can see here that albendazole by itself does absolutely nothing compared to DMSO. Paclitaxel does a little bit, but these tumors still grow essentially exponentially in this range, and we'll see even better in the next slide. But when you actually treat with the combination, you got regression, and then the tumor essentially works to essentially a stable state. The Kaplan-Meier is also significant, but we had to stop this curve here because the tumors started to grow so large that some of the subcohorts were started to have to be sacrificed, and so we couldn't represent all the mice. But if you look at the spider plots, you now see that the mice that were on the combo, 4 of the 6 had really striking responses that were very durable. And in fact, these mice were not sacrificed because the tumor was too large, but rather because they started to ulcerate. And so we're now repeating the study by first treating with paclitaxel, instead of treating first with albendazole and then with 3-cycle paclitaxel, a holiday, and then repeating indefinitely. We're now going to start first with paclitaxel so that we take down the tumor volume before it has a chance to do too much, and then starting with albendazole. I wanted to conclude this in the next 3 minutes with something we consider to be a really exciting study, because instead of targeting tumor cells, we're now targeting highly immunosuppressive cells in the tumor microenvironment. This is a collaboration with Chuck Drake and Alex Obradovich. So we isolated both peripheral and tumor infiltrating Tregs from 32 patients in 4 different tumor types. We asked, can we separate the infiltrating versus non-infiltrating Tregs by gene expression profile? Nothing, neither by tumor-specific type nor across all 4 tumors. However, if you do master regulator analysis, now you find 15 master regulator that are strikingly differentially activated in the tumor-infiltrating Tregs versus the peripheral Tregs, and an additional 2, 5 of these are in common, that are actually also specifically activated in the infiltrating Tregs, but not in the non-Treg infiltrating T cells, which obviously you want to maintain because otherwise you lose the immune response in the tumor. So to validate this, we did something that's called a CHIME assay. This is a chimeric assay where you do essentially a pooled CRISPR knockout screen of the 17 genes that we identified, plus a large number of negative controls, in hematopoietic stem cells, and then you use them to repopulate the bone marrow of a mouse that has been ablated by radiation. And then after about 10 weeks, where the immune system is completely rebooted, and now every cell, about 35% of the cells have a knockout of one of these proteins, you now implant a syngeneic tumor model. This is a very aggressive colon cancer model. And then you look at whether there was a difference in peripheral versus tumor-infiltrating T cells. And so we did this in two different cohorts. The second one with cells that were filtered with a reporter from the first cohort and then reimplanted. And you can see striking correlation between the results in terms of the profile of representation of the different master regulators gene targeting, the barcodes. And then you can see here, this axis here is extraordinary extended. So this top gene, TRPS1, has a statistical significant minor log p-value of 170. So 10 to the minus 170 is significant. And you can see that they're significant both in terms of peripheral Tregs, which were selected from the spleen, and also with respect to non-Treg CD4 T cells from the tumor microenvironment. And so we repeated the assay, but this time we only silenced TRPS1, which is a gene that nobody knows anything about in relationship to Tregs. And you can see that is quite a striking effect in terms of tumor growth in the 5 models with the non-targeting controls and 6 models with the targeting TRPS1. And in fact, if you look at the single mice, 3 of the mice had essentially complete response. The tumor was rejected de novo without any drug. And this is now with a different syngeneic model, because we wanted to make sure that this was not specific to that colon cancer model. This is a highly immunoresistant and aggressive sarcoma. So we then did exactly the same thing that we've done for the single cell in breast cancer. We asked, are there drugs that can invert the signature of the Tregs? And so we started with about 1,500 drugs that are FDA approved, selected the ones that do anything at all in Tregs, then did those response curves for those, and we selected the top 86. And these were now used to perturb tumor infiltrating Tregs selected from a kidney clear cell carcinoma, where you can get a lot of Tregs. And then we generated the RNA-seq profiles and predicted the drug activity. And what you're seeing here, the 3 drugs, gemcitabine, trypene, and floxuridine, were essentially able to invert computationally the signature of the infiltrating versus peripheral Tregs in virtually each one of the 32 patients that we had collected, except for maybe 1 or 2. But what was even more striking is gemcitabine was predicted to do so at 10 nanomolar. Now, gemcitabine typically is used in the micromolar range clinically and in PDX models. And so we want to know whether that was in fact the case. And in fact, gemcitabine is the only one that you can really validate, because it has a really gradual response in terms of the IC50, the dose response curves, where floxuridine has a very sharp elbow and then cytostatic, and trypene is really an equally sharp elbow. So you cannot really dose these 2 drugs in a way that they kill Tregs, but not the non-infiltrating Tregs or the ones, the cells that are involved in immune response. So this shows the response of tumors treated in vivo with 12 milligrams per kilogram, which is about 15-fold lower than what you would normally use for a preclinical study, which is around 150 milligrams. And you can see that there is absolutely no effect in the immunocompromised mouse. It's an NSG model, but there is a really strong effect potentiated by a PD-1 inhibitor. This is Nivo, that by itself has actually no response whatsoever without the treatment with gemcitabine. And you can see that if you go to 120 milligram, you now see the NSG model is starting to respond a little bit, but you still preserve the response in the immunocompetent models. So I'll stop here. Just wanted to say that, for the other people that are on this call that are not interested in cancer, this is not the way that cancer cell works. This is the way in which old cells work. And so we have used this model to essentially do studies in neurobiology, in many neurodegenerative diseases, cardiovascular disease. This was just published with the Riley Group. Type 2 diabetes, which just came out with Mimo Chili's lab. And then something that we're very excited about is the fact that now that we know how to do these analyses, we can literally take any cell state that you start from and any cell state that you want to end in and devise either a genetic or a pharmacological recipe to make it happen. And so, for instance, this is an example where we identified BAS2B as a single transcription factor that can reprogram committed human lineage progenitors into multipotent HSC state that was just published. And so just wanted to thank a lot of people and also the NIH for very generous support to this research. But this obviously is not happening just in one lab. It's a humongous number of collaborators that contributed to this and foundations. Also, everything that we do is made available to the community. Both our presented plasmid that we used are all available to Adgene. The software that we use is all available in Bioconductor or through our websites and our shared resources that generated all the high-throughput data. Thank you.
Fantastic. Thank you very much, Andrea. As usual, I've got a lot of questions. Let's see if I can stump you. But I'll just get started, and then please, anybody in the audience who has a question, do type in your questions in the chat, and then I'll repeat them. Where to start? So I've been convinced for a long time that you can find these master regulators. So as you know, we take a kind of much more traditional, I would say, approach to finding and developing new therapies, which also works, I would say. And so
works really well.
And so the question is, so what we've found, where I'm going with my first question, where we've found is, of course, what everybody has found in cancer is that single agent treatment of anything, I don't care what, is insufficient. Okay, it's there are exceptions, BCR able, you know, but they're rare. So we have gone to developing fairly complex mixtures of targeted agents. So my question is, computationally, how does that work out for you? Can you there's two different ways of thinking about it. You have two parallel pathways that control different things. You want to target each, or you want to target one very important pathway twice. So how does your mathematics show you the way forward, which is the best way to combine drugs in multi-targeted therapy?
So let me first address the question itself, because there's a little bit of philosophy behind it that I think needs to be brought to bear. There are really three fundamental reasons why single agent therapy fails. One reason is that most drugs have actually been built to target individual proteins that are actually oncogenes or that are involved in an oncogenic pathway. And so you're starting with drugs that are almost built to fail because, within the cells, we did the math, and you have more mutational patterns that can induce a tumor than atoms in the universe. There's about 2 to the 400 potential mutational patterns from the oncogenes that we already know about, and there's only 10 to the 80 atoms in the universe. So basically, every single cell that you have in the tumor will actually accrue additional mutations that will defeat a single cell therapy directed at an oncogene, right? So that's number one. Number two is that you have within the tumor different subpopulation that have completely different drug sensitivity. And this is actually epigenetically determined, not necessarily genetically determined. In fact, as I've shown you, you can literally spontaneously reprogram the cells from one population to the other without having to change their genetics. And the third one is that cells are incredibly good at performing what's called adaptive response. So they really rewire their networks in response to a drug. And this beautiful work by René Bernard that shows how, for instance, the MAP kinase pathway is rewired in response to BRAF inhibitors. So taken together, those three really formulate a manifesto failure of single agents, right? So what we have observed is that when you go and perturb the cells with drugs, you actually don't change the population. The population that are stable population that you had at the beginning before treatment are almost never changed. In some cases you do. For instance, in prostate cancer, you get a completely new population of neuroendocrine tumors cells when you treat with abiraterone and you have a p53 mutation, but it's very rare. And in fact, I showed you that even for the breast cancer analysis, when you treat two completely different drugs, the states remain the same. And so what we do is, instead of focusing on the pathway or focusing on the oncoproteins, we focus on the state of the cell. We essentially ask, what are the proteins that maintain the state, and how can we target them pharmacologically? Where this connects with what you do is that many of the proteins that you try to target are in fact the upstream modulators of the master regulators in the cell. So if you remember from that paper on NF-kappa B with Ricardo, we did this analysis in a kind of poor man way, and we showed that NF-kappa B was the reporter for the activity of all these mutations in its upstream pathways. And so when you find a drug that acts on BTK, you find a drug that is actually disabling the master regulators through a mechanism that it's complex, because there's a few steps to get there. So what we're pursuing, and now we're doing this in collaboration with a lot of pharmaceutical companies, is an entirely different collection of drugs that, instead of targeting these genes that are upstream, are actually targeting directly through PROTACs, for instance, or through antisense technology, are targeting directly the master regulator.
No, that makes sense for the future, for sure. So let me ask you.
In terms of combination,
yeah, bring it down
either you find drugs, either predict drugs that target different population and you combine them together, as I've shown, or you find proteins that are synergistic.
So, for example, let's take something I'm very familiar with. That.
Yeah.
So you have a lot of things that don't work as a single agent, but if you simply inhibit BCL2 in a lymphoma, then that's the ball game. That basically. And so I don't know whether you would refer to BCL2 as a master regulator. It is this master thing in the cell. So how does that fit in your philosophy?
Well, we published BCL2 as a master regulator a long time ago, and I think it was 2010. So it definitely comes up as a master regulator proteins, but
it's not a master regulator in terms of.
It's not a direct regulator of. Yeah, it's not a direct regulator, but what happens is that sub-signaling proteins are so close to the transcriptional layer that essentially act as co-transcriptional regulators, either because they directly phosphorylate or ubiquitinate a transcription factor. And so you can really transfer their activity almost as if it were a transcription factor. So the analysis is not as good for those proteins as it is for transcription factor. You go from about an 80% to about a 65% validation rate, but it still does really well. So for instance, that's how we found AKT1 as the master regulator of a glucocorticoid sensitivity in TLL with Adolfo, even though AKT1 is not a transcriptional regulator. So, but in general, we would say that probably BCL2 is implementing an even more transcriptional specific control module that BCL2 in some way inhibits in an amazingly good way. And so you're, by activating that, it'll work.
So I have one question from the audience that jives with one that I was going to ask. So the question was from Chunhua Yen. Sorry about that for butchering your name. And it says many cancer drug companion diagnostics are mutation based. How are the mutation information used in combination with the master regulators? And I'm going to broaden this out in general to say, if you were going for regulatory approval for a drug that's targeting master regulators or combination, what would it look like? What would be your diagnostic that would lead to identifying the response of patients? And how does that fit with what he or she asks about mutations as part of the diagnosis?
So we now have, as I told you, a CLIA certified assay that can be used to use the activity of any protein as a reporter for the activity of a drug. And so we have already published several manuscripts where we've actually produced predictors of the activity of drugs in patients. This is not in PDX bottle cell lines, including fimpinostat in the LBCL, including selinexor in AML, the LBCL, and glioma. We're now actually just got funded to start studying glioma based on that biomarker. And so I think Todd Gold's lab has published a paper on bioRxiv where it shows that among all the things you can use in the multi-omic landscape to predict drug activity, genetics is absolutely at the bottom. And I found it very refreshing to see that results, which we've been sort of proposing for a long time, came from a lab that is at the broad, but it's basically reality. And so the layer that is most informative one in his analysis was gene expression. What we've shown is that the protein activity is dramatically more effective than gene expression predicting. In fact, what happens with gene expression is that when you build a classifier, the more genes you use, the better the classification becomes. That's a classical sign of overtraining, overfitting. With protein activity, you basically get better with the first 6 or 7 proteins, sometimes with the first 4, and then it actually gets worse or it doesn't improve anymore. And so basically this tells you that that's the minimum number of markers you can use, and it's very good because clinically, using only 4 proteins is a great way to do it even in situ, instead of having to do gene expression and looking at a panel with a gazillion genes. So as I said, this is all published. I can send you the references.
No, it's fine. But I'm going to not let you get the last word on that subject.
Okay.
So what clearly individual gene mutations have their limitations but what we've been focused on, as you probably know, and it's related, I think, to the master regulator idea, is genetic subtypes. So this is not individual mutations. These are collections of mutations that seem to co-occur. And I'll bet you dollars to donuts then that when we define a genetic subtype, basically what we're seeing in an indirect way is a different class of master regulators that goes to that subtype. But in the paper we just published in Cancer Cell, these things have a 100% predictive power for response to ibrutinib or benefit from ibrutinib to ARCH. So it is maybe that we haven't been sophisticated enough about understanding how to think about these mutations, and maybe there's a nice
I think
there's a nice point in the middle that takes into account both of those bits of information.
I think there's a rationale for that, Lou. In fact, I think there's 2 things. First of all, your work, which is extremely sophisticated, could not have happened without very, very precise pathways, signaling pathways built by immunologists over 50 years. Okay, so you're actually benefiting from something that almost doesn't exist in other tumor types.
True.
Right? And so basically, you're doing what we are doing, but you're using the literature to do it in a way that is very informed. And you spend a lot of time. But you're finding exactly that this mutation coalesce into a pathway, and if you target that pathway, and now you know all the mutation in that pathway, you can predict. That's great. If you do it for a tumor, like for instance pancreatic cancer, where every tumor has the same main mutation is MAT4, P53 and KRAS, and the other mutation are just completely incomprehensible or mostly incomprehensible, then you really don't know what to do. You cannot use the approach that you have developed, Lou. And so I think this is where the common ground exists between what we do and what you have done. And in fact, to some extent, we have taken a page from your book, and the way we have started to develop these things is precisely.
Just for the audience, and we'll stop it there. When I'm saying a genetic subtype, I think you should sort of think of, luminal breast cancer versus basal. That's the kind of big distinction, and that does track with a bunch of different master regulator differences between those two. So it's not just one pathway; it's a whole constellation of.
It does, but
it's sort of like a cell of origin based type of cancer that seems to track with therapeutic response. And what I'd love to do with you collaboratively is to see if it tracks with master regulators. So We'll leave it at that. I hope this was, I hope the last question answer was entertaining for everybody. But thank you, Andrea. I always learn a lot, and this is another great lecture. Thank you so much.
Thank you, Lou.